Hello and welcome to another screencast where this time we'll be continuing the discussion about signal transduction pathways. And this time we're going to focus right here on describing and explaining the elements of the signal transduction process. So this is the transduction. We'll talk first, actually this is in reverse order, sorry. First we'll talk about the second messengers and then about signaling cascades. Okay, so as we had seen in previous screencasts, we have this first messenger, or the hormone signal molecule right here, right? And they're giving you an example as epinephrine here. That first messenger is going to bind with the receptor, so this is the first step in this. This happens to be a G protein linked receptor, and this is all part of that G protein linked process right here, where the G protein becomes activated and then binds with this adenylyl cyclase molecule here. So then we said that that adenylyl cyclase molecule is going to become active. So this thing right here is an active enzyme. And that enzyme is going to catalyze the production of this second messenger called cyclic AMP or CAMP. Right? So this is cyclic AMP. And we see that's what we see over here. So we're going to start with ATP, and we're going to, in the presence of adenylyl cyclase, so this is the enzyme adenylyl cyclase, right, is going to take two phosphate groups off of here to form this pyrophosphate thing, right? And so that's this right here. Right? That's what you see right here, where there's only a single phosphate. So this adenylyl cyclase, to shorten this up again, the adenylyl cyclase is taking these two phosphates off of ATP. You make this cyclic AMP thing right here that's then be hydrated, and you then get AMP. Now, what's the big deal with this? Well, this cyclic AMP molecule right here is, pardon me, is active, is reactive, right, and will act as a second messenger. So this second messenger is that it's not the first messenger. So this is not extracellular, it's not a hormone, it's not a chemical outside the cell, it's intracellular. Right? And so this is another molecule, this active cyclic AMP thing here, that will then activate this protein kinase A. Right, now, we talked about this in some sections, but maybe not in others, but a kinase enzymes are involved in phosphorylations. Right? And activating other molecules by phosphorylating them. So essentially what we have here is one enzyme catalyzing, so one enzyme catalyzing the reaction, the activation of another enzyme, etc., via this second messenger, right, or this cyclic AMP thing here. Right? So without this cyclic AMP, you wouldn't get the activation of this protein kinase. So in this way, the signal is being transduced. We're changing it from just this molecule that's on the outside of the cell to this whole series of phosphorylations, right, involving GTP, involving ATP. So every time we phosphorylate something, we're of course making it to a higher energy level. We're changing its reactivity, making it more reactive. Right? And so the cyclic AMP is an intermediate molecule that will then activate this protein kinase A. So that's one example. A very common example of a second messenger is cyclic AMP. So pause there and sort of review that, see where your questions are. Another example of second messengers is this diagram, which is kind of complicated. So let's start with what we know first. Okay, so here we have this first messenger again, binding to this G protein linked receptor, right? And then this G protein becomes activated here, right? And then this G protein is now going to activate this phospholipase C enzyme. Right? Phospholipase C will cleave this yellow structure, right, that is attached to a protein that's in the membrane. Right? And so when this phospholipase C cleaves this, it cuts that, right, and what this thing becomes is IP3. This is another example of a second messenger. Right? What's left behind is the DAG thing here. Right? So DAG plus IP3 is PIP2. So this PIP2 thing is equal to DAG plus the IP3. So when we go this way, we release this second messenger IP3. When we go back this way, we just make this PIP2 thing here. So the enzyme phospholipase C is catalyzing this forward reaction this way of changing the PIP2 into this IP3. What's the big deal? The big deal is that this is now another example of an intracellular molecule that will then go and do things. It turns out that in this case, this IP3 will bond to a calcium channel that's in the endoplasmic reticulum here. So when this second messenger here binds to this channel protein on the ER, right, that actually opens up the channel, and then calcium ion is able to diffuse down its concentration gradient into the cytoplasm from the lumen of the endoplasmic reticulum. So this endoplasmic reticulum is a storehouse for calcium, right, ions. Turns out that this calcium ion being released into the cytoplasm itself is also an example of a second messenger, right? And for various reasons that we can talk about in class in detail, is that this calcium ion will activate various proteins, right? Proteins meaning like enzymes, right? If you are a student of anatomy and physiology, then you know that calcium ion will be involved with activating actin and triggering its reaction with myosin to get the muscle to contract, right? Other examples are that calcium is going to trigger, say, the breakdown of glycogen here, right? So this is now a response, right? So this would be a response, muscle contracting down here. This would be a response here, right? Glycogen being broken down into individual glucose molecules, right? Students of anatomy and physiology know that this is glycogenolysis. Right? So this glycogenolysis is triggered by, pardon me, is triggered by this calcium release here. Glucose is then exported from the cell, and then so on and so forth. So this is an example that we're going to see in detail when we look at blood glucose regulations. Okay? So pause there a second and summarize that. See where your questions are. A different transduction model here is not necessarily using second messengers, but is using more of what is known as a phosphorylation cascade. So again, let's go back to what we know. Here's the signal molecule, the hormone binding to the receptor. Right? Now, in this case, this receptor isn't a G protein-linked receptor, isn't a tyrosine kinase receptor. They're sort of generalizing this a little bit more. But what's happening is that this activated relay molecule here, right, was assumed to be inactive. When the signal molecule is not bound to the receptor. So this activated relay molecule is going to take this inactive protein kinase here and make it active in some way. This active protein kinase now, again, we said that kinases are involved in phosphorylation reactions. So this protein kinase here, right, will phosphorylate inactive protein kinase 2 and make it active protein kinase 2. Now I want to point out right here that this is what this looks like, right? That this is the active protein kinase and it's been phosphorylated, right? This is the inactive protein kinase enzyme and it is not phosphorylated. So by hydrolyzing ATP here to ADP, we take this inactive, make it active by adding a phosphate group onto it. Well, this active enzyme right here that's been phosphorylated catalyzes the reaction of this inactive protein kinase here by transferring phosphate groups. So this phosphate group, this gets broken, right? And then you get this inorganic phosphate going off. That is an exergonic reaction that catalyzes this endergonic reaction. This exergonic reaction then, right, catalyzed here, right, to get this endergonic reaction, which is the phosphorylating. And this keeps happening. So you see how this is a cascade, right? Or maybe you don't, but you should try to see how this is a cascade of reactions. So this starts the cascade, and once you get this activated relay molecule going, you then activate a protein kinase, which activates another protein kinase, which activates another protein kinase. So in this way, it's a cascade. Lastly, this cascade has to end somehow by this active protein kinase here phosphorylating this inactive protein by a similar mechanism, and then you get this active protein here that's phosphorylated, and that's going to lead to some sort of cellular response, which is varied depending on the cell. So instead of using second messengers here, we're using a phosphorylation cascade as the transduction, right? So the signal has been transduced, it's changed its form, right? That ultimately what you're trying to do is get this activated protein down there, and you do it via protein kinase cascades. An advantage of this is seen with an example of these cascades, that they often will amplify the signal. So you have a very low, here we have the relative concentration, right, over here. So one molecule of epinephrine, right, also known as adrenaline. So one molecule of adrenaline, right, binding to a G protein linked receptor will trigger this phosphorylation cascade. And this phosphorylation cascade, you can see here, is being amplified as we go on down the line here. So all of these enzymes here becoming activated, right, will amplify the number of molecules down here that are activating this glycogen phosphorylase. And this glycogen phosphorylase is the enzyme that is breaking down glycogen into glucose 1-phosphate. So from one molecule of epinephrine, we have 10 to the 6th molecules of this activated enzyme. So you can see here that very rapidly that cell that is going to be signaled by epinephrine will very quickly make glucose available to the cell, right? In fact, it makes 10 to the 8th molecules available to the cell. So it does not take a lot of this signal to get much of a response. So of course, graphically, this image looks like this, and you get this sort of response here, exponential response there. So that is an advantage of using these chemical signals, and that is a characteristic of many transduction pathways within the cell, right, is that you amplify the signal. Okay. One final example here. Again, we see some sort of growth factor, right? So some sort of growth factor hormone out here binding to a receptor, setting off this transduction. This is the cascade. And it turns out that then this active protein kinase here will actually enter the nucleus and activate a transcription factor, which then starts the transcription and translation of this actually translating outside here in the cytoplasm. So this is a way that external factors can regulate gene expression. Right? That this external growth factor here will actually turn on, right, or very well could turn off the expression of certain genes in the cell. Okay. So what we've done here is talked about elements of the transduction processes here. We started with these second messengers. Cyclic AMP was one, that IP3 was another, and calcium ion is yet another. So looking at some specific examples there. And then characteristics of signaling cascades in terms of understanding what a kinase enzyme is going to do, right, as well as then amplifying and the application of that, either activating proteins within a cell or actually regulating the expression of genes. Okay? Thanks for listening. Bring your questions or confusions to class.